So cells are the smallest known living things, and they arise from pre-existing cells, and all cells are related thanks to evolution. Now let's look at the scales we're talking about. Here is a scale up here at the top is 10 meters, and down here is 0.1 nanometers. So you can go back to that slide I showed you a little bit ago to get a sense for what we're talking about here. But 0.1 nanometers is the scale we might use to measure, an atom or something. Now, if we go all the way up here, this shows us the unaided human eye, what it can see. And the unaided human eye can see nerve cells, if they're extracted, because they're really long, chicken eggs, which are a single cell, just a highly modified cell, frog eggs as well, and we can just barely maybe see a human egg, which would be like a dot, basically, to us. But starting down here, actually starting around here, you can see where light microscopy comes in. These are the kinds of microscopes we would use in the lab, right? We can use those to look at frog eggs and human eggs, and even look at plant cells and animal cells and bacterial cells and some organelles like mitochondria and some really small bacteria down here. Okay, we can see quite a few pretty small things. Then if we want to see things that are really small, we might start using electron microscopy. Electron microscopes basically shoot a beam of electrons at an object, and then the electrons bounce back off and an image is formed. And we can see big things like cells with it, but we can see even smaller things like proteins and lipids with it. Okay? So electron microscopes will let us see things that are even smaller than we can see with light microscopes. And we have a scanning electron microscope in the department, and you can take a course in how to use it if you wish. Just to give you an example here, here's some images made by light microscopes. Here is a microscope like you might use in labs. This is probably somebody's cells from their cheek. You can swab your cheek and make a microscope slide of it, but they're hard to see. But then you can add stains, and stains let you see the cells more clearly, as seen there. So there's something you could easily do. You could easily look at those in lab here in our department. You could get images like this. But then there are other fancier scopes, like some of the cell biologists in our department use fluorescence, that you stain cells so that different parts of them glow so they really show up. But all of that is light microscopy. If you want to get really close, you use electron microscopy. So here's an image here with looking at cilia with a scanning electron microscope that bounces images off of the outside surface of an object so you can see it, see that organism. Or transmission electron microscope, or TEM, that's when you slice the organism very thinly and you look at its insides with a electron microscope. On the left, I thought I'd just show you, I study paleobotany, fossil plants, and on the left here we can see this is a piece of a flower of a fossil sycamore tree from the Cretaceous period, the time of the dinosaurs, and you can see that it's loaded with pollen grains, and here's what they look like under light microscopy. But if we put them under scanning electron microscopy, you can see the scale bar here, that's 1 micron, you can see all the detail there that you can see on the external surface of that pollen grain. Now, if we were to slice that pollen grain and look at its structure that way, that would use transmission electron microscopy. So, what are the structures found in all cells, whether you're a eukaryote or a prokaryote? This is something I would ask you to answer out loud. So see if you can think of an answer before I say anything, because that's how you pass a test. All right, a couple things. Hopefully I don't skip any, but you can read your book and find these as well, right? I expect you to read your book and emphasize and re-emphasize this and learn about things I don't talk about. But the first thing all cells are going to have is a plasma membrane. That's the membrane that surrounds the cell. The other thing that all cells, whether eukaryotic or prokaryotic, are going to have is the cytosol. This is the liquid inside the cell. Another thing all cells are going to have is DNA. Probably DNA and RNA. I should just say genetic material would be even better. But let's just say DNA / genetic material. And lastly, what do you need to go from DNA to protein? You need ribosomes. Ribosomes are made of RNA. Remember, they have that kind of sandwichy structure like that. And they are used to make protein. So you'll find all of these structures in both prokaryotes and eukaryotes. Now, a prokaryotic cell is a pretty simple cell, and there's a great diagram in your book, but I'm just going to draw the bare bones. This is a one-room cabin, basically. Here's the plasma membrane. We'll talk about plasma membranes in the next chapter in greater detail. It might have a flagella or something out here to help it swim. It will probably have some kind, I'm just going to draw a little bit here, some kind of cell wall. Most of them do anyway. I'm not going to draw it all the way around because I don't want to take all day. And maybe a few other things that you can learn about in future courses like microbiology. All right, inside you're going to have a ring of DNA called the nucleoid. Right? And you might also have smaller structures in there that have DNA as well, like we've talked about in the past, those plasmids. And then you're going to have ribosomes in there. Okay? So those are some of the things you'd find inside of a prokaryotic cell. So in the next video, we're going to pick up from there and we're going to look at what we would find in a eukaryotic cell, and more specifically what we would find in a plant cell and an animal cell.